it is Sully and Jones, and we are here today with Kitchen Table Research, where we do the research so you don't have to. And we have put a couple of episodes in between season 1 and season 2. The first one was the Max DeVries case. Unfortunately, Max went missing in Aruba, and if you'd like to learn more about his story and what our audience can do to help this family, go back and listen to that episode. We just felt like this episode needed to come out because of its importance and what the family is trying to uh, basically get the FBI to be involved in this case. So go back and listen to that. It's on Spotify, YouTube, wherever you listen. And then today we are kind of presenting an in-between case of the Golden State Killer. But today we're really looking at the DNA because as we go forward in season 2 and especially into the Oklahoma Girl Scout murders, DNA becomes very important. And as you can tell in season 1, DNA was also very important in the Darlie Routier case.
Well, and it was also important in the Michael Morton case because it got him off.
It's really important in every single case we did, even going all the way back to the yogurt shop murders.
Even though it's not been matched, there is DNA in the yogurt shop murder case.
And that's kind of how the Oklahoma Girl Scout case is too, correct?
Correct.
Okay, so before we dive into DNA today, let me shout out all of our channels. So we have grown rapidly since season 1. We still are on Facebook and Instagram under Kitchen Table Research, and we are starting to put all of our content and build up those channels for ourselves. We have everything on YouTube, and we also have some exclusive interviews and exclusive content on YouTube. But if you really want to follow us, jump onto TikTok. You can find us under Kitchen Table Research or Marcus the Bunny, where we have exclusive content including medical mysteries. Yeah, we're just trying to expand, and I have another amazing, I don't know if I told you about this, Jones, but I have another amazing update. So we finally have our first official sponsor for the podcast, for our TikTok, for everything that we do, and it is this moisturizing face cream from the company GoPure. They make a lot of great products, and any of the commission that we make off of our GoPure products not only keeps the podcast ad free, but we are also helping families that have been affected by crime. So if you want to go shop on our TikTok, it does families and our channel. It's very good for both. And I just want to tell you, this Titan and neck lifting cream it goes on sale for about $30 on TikTok. It has the Cacao butter, hyaluronic acid, and caffeine. And for the last 6 months, it has removed dark spots, even my skin, and even taken away some of the wrinkles around my eyes, my smile lines, and my forehead. I know, Jones, you've tried this product as well. What do you think about it?
Well, I like the product. I just can't seem to manage to get it between the two houses that I live in. If I want to use it, it's at the wrong house. If I'm in Carville, it's at the wrong house. And so I like it. It goes on smooth. It's not an obnoxious smell.
And it really feels like it penetrates.
Obviously, this wrinkle right here has not faded as much as this wrinkle right here. Yeah. But I like the product.
Me too. I really like it. They make retinol, vitamin C. This is like their basic moisturizer. I would say definitely go pick it up if you're looking for a good moisturizer. And like Joan said, a lot of moisturizers I feel like sit on top of my skin. I can feel this one actually like being absorbed by my skin because of that Masterburst hydrolyzed hyaluronic acid. I think the droplets are smaller and it really gets absorbed by the skin.
It's not so much the droplets, it's the molecules.
There you go.
Okay, so the smaller the molecule, the more likely it is able to be absorbed into your skin. So hyaluronic acid is a big one, is a big molecule. Mm-hmm. Vitamin C is a big molecule. So you're looking for baby molecules in those products.
And that's what GoPure has. So
I do need to ask the question, and I've asked it before. I'll put you on the spot here. I have tried repeatedly to pull up. What's the one that the iTunes podcast.
Yeah, you're having an issue finding it on your Apple podcast.
I am. I cannot find any of those podcasts that we did.
Well, we'll talk about it after this.
Okay, well, I really like the iPad podcast format. You know, I'm not a TikTok girl. That's just not the way I like to listen. I like to listen in my car, and I like to listen before I go to sleep.
I think there must be just some kind of weird thing where you're not able to find kitchen table research right now, but I guarantee you it is out there, and when we get off this call, I will show you exactly how to go find it.
Yeah, right.
But if you are on Apple Podcasts or Spotify, just type in kitchen table research, and you should see us come up. So it may just be because we haven't released an episode in a little bit that it's kind of been pushed down in your algorithm, but we'll get it figured out. And everybody, all of our fans, if you guys are having issues with this, please email us at kitchen.table.research@gmail.com. If you're having problem finding any of our content, or if you just want to say hi, we'd love to send our fans bumper stickers. So that's a great way to get a hold of us one-on-one, is by going through email, which is kitchen.table.research@gmail.com. So, okay, are we ready to dive into DNA?
Yeah, and I did a lot of research on the DNA, and I stay confused on DNA. I'm better than I used to be, but Jones had asked me to research the Oklahoma Girl Scout murders. They happened in 1997. And when I was starting to familiarize myself with the murders of these young girls, it became obvious that the DNA knowledge in 1977, when these murders occurred, was minimal, and it's really outdated now. I mean, we know so much more in the past 40 years than we did in 1977. They did try to collect DNA. It was quite minimal. They really didn't collect any DNA from the crime scene. They I think, collected a latent fingerprint that they could not match back to the man that they were looking at for the murders. And so that case became unsolved as of this point. So I decided I was going to try and re-explain DNA and focus on what we know today as consumers about the current knowledge in DNA profiling. As I said, it wasn't even on the radar as a forensic technique until after 1977. And I talked to our correspondent over in England, and she said the first legal case that was in Britain was not until the mid 1980s. So the technology on DNA grows by leaps and bounds at least every year, if not every day, because I read a lot on DNA and I always find out something new. Or maybe I should say it's better explained and I understand it better.
I agree. In my research of the Golden State Killer, which we have followed this case for so long, and we've used it as an example because it really is a precedent case in DNA. So that's why we're using this case as an example today. Also, it was worked by Paul Holes, who, in my opinion, is like one of the best investigators. Like, I wish Paul Holes would investigate Darley, you know what I mean? But in my research, it was America's first case was solved by DNA in 1986. So, about the same time, mid 80s.
Right. And then, of course, you have to take it into a courtroom and convince the judge and jurors that it's a valid, appropriate, well studied method to identify people.
And I
think, a killer or a victim.
Especially in the beginning days, I think it was, I mean, we're still having a problem understanding it. Obviously, the techniques and the intricacies have gotten greater in the last few decades. But if I was sitting on a jury in 1990, 1996 per se, you would really need somebody, an expert, and then a layman to explain what that expert is actually stating. You know?
Yeah, you know, my background has always been in pediatrics, and I tend to, I don't want to say, I tend to talk to the children. I don't tend to talk to their parents. And so when you need me to explain something, I can get it down to really a low grade, even toddler level so that they understand what I'm trying to do. This goes right over my head sometimes.
Yeah.
And the terminology and the reallocation of words as they've discovered more just changes on a day by day basis. So today I want to discuss the evolution of DNA in the United States. And then I want to get into the Golden State Killer because he was identified and arrested in 2018 after only 4 months of work by a 5-person investigative team. That is huge.
And
the Golden State Killer started his spree in 1975, I believe. So in 2018, what is that? 33
25.
years later, if I can add right. And,
yeah, almost 40
they had,
yeah.
Yeah, they had just figured out that there was a way to track people that we had not been using. And so that's kind of the story. It's kind of the story of how that all evolved.
Well, and the documentary that I watched, which I'll give you guys the name of it here in a bit when I get to some of my talking points, but Michael DeAngelo, who is the Golden State Killer,
Joseph DeAngelo.
I'm sorry, Joseph. I can't get my names right today. Joseph DeAngelo, who is the Golden State Killer, his DNA had been in CODIS since 2011, 2001. So why did it take, let's see, since 2000?
Why was his DNA in CODIS?
Well, I mean, they had the DNA that they had taken from the crime scenes because not only was the Golden State Killer obviously a killer, but he was also a rapist. So they were able to get samples of DNA, but they had no one to match it to. So in 2001, Paul Holes and his team put Joseph DeAngelo's DNA into the system. So why did it never match? Well, because DeAngelo was never picked up for a crime.
It takes 2 people to do a match. Okay, it takes the perpetrator, who was Joseph DeAngelo, and it takes one of his, and it takes the actual person, Joseph DeAngelo, to make that match. And I'm going to get into that here in a minute.
Okay, perfect. But I just thought that was so interesting that they had put his DNA in there in 2001 and it sat there for what, 17 years before it matched? Okay.
Right.
So explain to us how did they get there?
Okay, well, let me kind of start back up. We've talked about DNA before in season 1 of our podcast, in particular on the Michael Morton case, because it was really the DNA in that case that exonerated him. We knew that we knew that something existed in the early 1980s that was unique to each individual. And we ended up, I don't know how it got its name, I'm sure it's some chemical formulation, deoxyribonucleic acid, shortened to DNA. The entire DNA project was completed while Bill Clinton was president. Well, Mr. Clinton's still alive, so you can tell that we're not far along in the discovery of how DNA works and how we can use it to scientifically prove things. So I mentioned a minute ago, it changes on a very regular basis, and it is difficult to stay up.
And didn't the people that mapped the entire DNA genome, they won a Nobel Prize for that, if I'm not mistaken?
Right.
Okay.
Right. This was not silly stuff.
Yeah, this was a massive discovery in the 90s.
Massive.
Yeah.
I know you guys can't see this, but I think everybody, I have a picture on my screen, but I know everybody can't see this, but most people are familiar with the spiral helix.
Yep.
Okay. And that spiral is made up of phosphorus. Okay? An ordinary thing that we make in our bodies. Okay?
Mm-hmm.
In between each of the spirals, there's a base pair of things that match up with each other. Okay?
So if you think of a ladder, and I know it's a helix, but if you just think of a ladder and think about every rung, two pieces make up every rung.
Right.
Okay.
And those are adenine, which only pairs with thymine.
So AT.
And ATs, A and T's go together. And guanine pairs with cytosine. And there's a little bridge between each of those base pairs, and that's made up of sugar. Now,
okay.
Everything that we know of, even plants, even animals, follow that same algorithm as well.
Correct.
So we all have this helix that twists around each other. So if you want to know if the skunk that's dead on the road came from parenthood A versus parenthood B, we can tell you that now.
Right.
It's used fairly extensively in crop growth.
Mm-hmm.
It's how we've gotten better crops and more yield from our crops, so that it doesn't take as much land to grow crops. So there's a tremendous amount of value that's come out of this exploration of DNA.
And I know a lot of people are like for and against GMOs, but as you can tell, you might hear the word GMO thrown around, but I don't really think the massive population understands the pros versus the cons of GMOs. So
correct.
Yeah.
Of manipulating the DNA to get what you want. And that was always a question. Do you remember when they were manipulating the sheep named Baby Dolly?
Oh, yeah, Dolly, yeah. She was the first clone.
She was the first one, and they were trying to manipulate her DNA to get the kind of sheep that they wanted. So she was probably bigger. She could probably carry more babies. But we've been jacking around with DNA for quite some time.
And it's been quite controversial, like,
Absolutely.
Dolly, GMOs. So some of this is quite controversial, but I think in the and it's actually, we're going to get into the controversy with DNA inside of solving crimes by the end of this podcast.
Well, and I think it's kind of important to say, I forgot that train of thought. Never mind.
I'm sorry. Okay, continue.
That little girl that I birthed talks about me. So each helix carries a chromosome, and there are 22 of them. And those helixes join together. There are 22 of them, and it determines the individual that we pop out of our mom as. The 23rd chromosome is called the sex chromosome, and it determines whether you turn out to be a female, which is an X chromosome, or a male, which is a Y chromosome. So if you have two XX chromosomes, you're a female, and if you have an XY chromosome, you're a male. So that has helped filter out 50% of the people in the criminal world that we're looking for. Immediately you can say, well, this is an XY, and so the females are out.
Right.
And exactly the opposite. So that was a major find too, is when we found out that the Y chromosome makes you a male.
And I think we've known that, like, pre-DNA, they could do sex typing and blood typing.
Correct.
And that was kind of the extent pre-DNA of what they could do. And I think we'll get into some of that when we do the Oklahoma Girl Scout murder. So.
Right. And it also allowed us the ability to give people blood.
Yes.
Okay, like an accident victim. In the early days, we may have given them blood, and they may have had a reaction from that blood and died. And nowadays we know enough to say, if you're an AB blood type, then I have to give you AB blood. That's not 100%. There's a lot of exceptions to it. The most interesting, I think, is there are universal. Let me take that back. When I did transplant, if you're in the first 3 months of life, you can take pretty much any blood type,
Right.
and your body will make it what it wants to make it, which would be the individual's blood type. Okay. And I think I've mentioned this before. My husband has, I don't even know what his blood type is, but I know that they love it when he donates blood because he has some special blood type that has some special antibody on it that only babies can have.
Yeah.
And so they always jump up and down when he comes in to donate blood. So this is not hard and fast 100%. There are some variables, but this is kind of what we know.
Did you
Did
you know that your other child has that blood as well? Megan she gives blood all the time because of that.
Because she's what,
a universal
donor?
Yeah.
Randall's more than a universal donor.
Yeah, I think she's like that one that,
right.
But he's like even a step above that.
Okay.
Okay. So our family has good blood. We have good genes.
We have good genes. Well, we have good blood at least. So let me go back. And kind of explain. So those base pairs that join up, an A and a T, a G and a C, there'll be a whole clump of them. And depending on the way they're clumped in that helix, it depends on where they are in the chromosome helix, okay, depends on, am I in the eye color portion of that chromosome? Am I in the skin color portion of that chromosome? Am I in the long first toe portion of that chromosome?
A heart valve. I mean, every single little thing. The way your veins are constructed throughout your body, those are all sequenced in your DNA. So
that's right.
We use eye color and hair color as an easy example, but it goes your brain structure. I mean, we know that medical illness is genetic, so that's going to show up somewhere in your, like you said, it's going to show up somewhere in your DNA strands.
Right, and so, the hunt is on to be able to go in and alter DNA to get rid of diseases that we know are fatal.
Right.
Okay, so let's take cystic fibrosis for an example. It doesn't take long for a baby to be diagnosed with cystic fibrosis. They have a lot of respiratory issues and blah, blah, blah. But what about if we could go in and manipulate that portion of the chromosome, the helix, to fix it so that they never had cystic fibrosis? I mean, what an amazing feat that would be. And we're getting close. We are getting close.
And my question is, like, say we take out that gene or manipulate that gene that causes cystic fibrosis, your body is going to automatically replicate that DNA. So how much of the DNA needs to be manipulated for the body to start only reproducing the modified version? Right. So I think that's where you get into some of the nitty gritty.
They're called steps. Like when you take out this helix and the body may naturally snip it, okay? And it may do that when you're still in utero because it knows that there's something wrong.
Mm-hmm.
And the body goes in and takes that portion of the chromosome out.
Yeah.
So what if human beings, scientists, could manipulate that to get rid of what we want to get rid of?
Right.
Interesting, fascinating concepts, really.
Yeah.
So the lizard, let's talk about lizards for a second because they have the same helix structure that human beings do. And the lizard eye color is found in the same part of the chromosome that human eye color is.
Okay, so it's found along the same part of the spiral that it would in
and
humans.
And that was a big find, as we started studying where is eye color located. And well, let's say you're albino and you have no eye color. Could you snip in green?
Right.
Could you snip in brown eye color?
Right.
And make that person more, I don't want to say normal, but normal, at least not with those blank eyes.
Yeah. Yeah.
So I found that very interesting. So then we've also got something that's called a phenotype. And those are the sections of the helix of the chromosome that are influenced by outside forces.
Right. So this is where we come into the nature versus nurture argument.
It's nature versus nurture. It's things like environment. Okay, I get that.
Yeah.
Nutrition. I get that. That's why babies need, all the stuff that breast milk provides, all the stuff that formula provides. And interestingly, trauma can reshape the chromosome.
Yeah. Cellular memory can carry trauma.
Yeah.
So I could be carrying the trauma of our whole family's lineage on my back. Thanks, guys.
Well, I did not get that to you.
Oh, that came from the other. That came from the other source of DNA that I have.
I really feel I do not feel like there's some incidents I can remember that are traumatizing, but I did not come up through trauma.
No, I would say no.
I was not abused. I was not starved. I was not.
I've had traumatic events happen in my life that have changed my life, but it wasn't in the home during my childhood. Does that make sense?
Right.
Yeah.
Yeah. Yeah. Well, there are certainly things that are imprinted on my mind that I hope I never have to repeat again. So there you go. So really, when you look at a chromosome, you would originally think that it's a pretty rigid structure. And we found out that it is not a rigid structure. It is fluid.
Yeah.
But there's things that change it. Okay. So hair color. We've talked about eye color. Let's talk about hair color. Is consistently in the same region of the chromosome. But it's the base pair alignment that varies. So a redhead base pairing is different from a blonde hair base pairing.
Right.
And interestingly enough, I don't know where this, I just know this. Redheads are either easier or harder. I think it's harder to sedate.
Yes.
And put under anesthesia.
Yep.
Than a blonde, for instance.
Yep.
So if you're a redhead and you're laying on the operating table and the anesthesiologist is going to try to put you to sleep, he knows that by looking at your hair that he may need to use more or less of his anesthesia.
Yeah, they have an algorithm for anesthesia for redheads because you're right. They don't go under as easily as another.
They're harder to sedate. So I put in my notes an example of a base pair DNA sequence. A goes with T, C goes with G. So, here's one. ATCG, ATTG, AGCT, CT, AGGCT. That's the strand. That's one section.
One section.
Yeah.
You can kind of think of it almost as like binary. Like you can code infinite things using ones and zeros. Think about the infinityness of having 4. You know what I mean?
Right.
Yeah.
And you don't think 4 is that many.
Right, but in different combinations.
Over 33, I think it's 300,000 possible combinations.
I think that's what I heard.
Of base pairs, you understand why. First of all, you understand why we all look different. And second of all, for those people that look exactly like their mother or exactly like their sister, they've obviously got more base pairs in in
Tandem repeat.
in tandem repeat. That's a good word. Than my 48th cousin twice removed. We don't carry much of the same DNA.
So listen to this. This was I watched this on YouTube. It was really good. It's under Veritaslum. So Veritas, like the Italian word for truth, Veritas,
Right.
LUM. And it's they interviewed the lab and Paul Holes and Billy Jensen, who all worked on the Golden State Killer. Um so they're saying now, originally they were looking for those tandem repeats, right? To see, me and you are going to have more tandem repeats than me and my husband, right? Because we're a direct, especially because we're mother and daughter. So we're going to have a lot of the same tandem repeats. So they used to look at the tandem repeats like you're talking about this ATCGAT, GAGCTC, right? Well,
Yes.
when you remove all of the tandem repeats, you get left with what they call SNPs.
Those are SNPs.
It's called SNPs. Um, I was looking for what that stood for. Um, mm, mm, mm. Single nucleotide polymorphisms, or SNPs, which they call SNPs. So because humans, we share 99.9% of all of our DNA, we're going to have so many common tandem repeats, right, with each other.
Correct.
So take away all the repeats, and now they're just looking at the SNPs. So instead of looking at all the stuff that's the same, you throw that out the window, and what do you have left over? Well, you'll have an A in a random place, or a T in a random place. And actually, I have blue eyes are considered a SNP because they are sprinkled throughout the tandem repeat. So like you said, you have a whole block of this ATCG, ATTGAC, okay? Well, those are the same, so let's throw those out. And then in the middle, you're going to have these random letters. Now they're interested in those random letters, the SNPs. So that's kind of the newest. Once we figured out the ACTG tandem repeats, then they figured out looking in between them might offer more information through the SNPs. So they're sprinkled throughout the tandem repeats. Like I said, me and you are going to have almost identical tandem repeats because we're mother and daughter, and I mean, we look very much alike. But however, we're going to have slightly different SNPs. So like I'm like, we have the same eye color, we have the same hair color. Let's just say that my nose is a little bit larger than you. I have my dad's nose. So that would be a SNP that would be different than yours. And so now that's what DNA experts are looking at, are the SNPs, not the tandem repeats. Does that make sense?
It does make sense.
And blue eyes are a SNP. Blue eyes are something that they can see quite easily now as a SNP.
So, DNA is not on everything. It's really, I think, the easiest way to think about what might DNA be discovered in is fluids. Okay, blood, saliva, semen, secretions, sweat.
Yeah, okay. It's like the carrier.
Yeah. Huh?
It's like a carrier.
Right. And we're going to come into that here in just a minute as we get into the GSK case. So the second way that we have learned how to examine a person's genome is familial genealogy.
Yes.
And and that's what was used to capture the Golden State Killer.
Okay.
So by looking at the genes as a group,
Yes.
or their phenotype or allele, another fancy word for a clump of DNA.
And an allele is just one binding, one rung of the ladder.
Yeah.
Okay.
And drawing inferences. Okay.
Mm-hmm.
So if family members all group as Cuban,
Right.
let's say, geneticists are going to first examine Cuban in their hunt for relationships.
Okay. I'm there.
Okay. I read a couple of books when I was doing my research on Oklahoma Girl Scout case.
Uh-huh.
And they talk about markers.
Right.
Well, that's a completely new word to me. When you're looking at current terminology being used in DNAs, I couldn't find markers. So what I think is a marker that they used back then is now probably known as an allele.
Or a snip.
An allele is a section
Right.
of DNA strand that is particular to an individual.
So that's
my
interesting.
allele for green eyes might be the same for you, or it might not be the same for you. And maybe we're off by a base pair.
Right.
So when the DNA was applied to the prime suspect in this Girl Scout murder, 3 out of 5 known markers on the DNA strand matched him. Okay, well, what does that mean? Well, we know that the number 1 marker was that it matched as Mongolian. And so that's that whole race of people that came across from Eastern Europe through Russia. They walked across the Bering Strait.
Was that like the Genghis Khan? Like so many people carried his DNA because of that movement?
Because he impregnated lots of women.
I mean, this is back in the rape and pillage era.
Yeah.
Okay.
Yeah. And so the Mongolian match includes American Indian. And the prime suspect in the Oklahoma Girl Scouts was actually Cherokee.
Right.
And that types him as Mongolian. The second one is it matched for male. Okay? So he had a Y strand. The third match has never been revealed. If somebody knows that, I don't know it.
Do you think it's like hold back information in the case?
Correct. I think there probably was, but they didn't even know enough to hold back then.
I know. Yeah.
So he matched. Back then, you had to match. There were 5 known DNA markers, and the suspect matched 3 out of 5. Okay.
Mm-hmm.
And that makes a 60% match. Got it? 3/5 is 60%.
Yes, I'm with you.
And that makes it an inconclusive match.
Right.
An 80% match would have been conclusive.
Okay.
But they don't have that. They've only got a 60% match. So everything about this man that was tried for the Oklahoma Girl Scout murders was inconclusive.
Okay.
That we don't have enough. And, oh, by the way, they didn't find a whole lot of DNA. I don't think they found any.
Well, it's degraded, blah, blah. But we'll get into that when our next case is going to be Oklahoma Girl Scout murders.
I'm going to start in on Golden State Killer.
Yeah, let's get into the Golden State Killer. I'm excited for this.
Oh, no, I found this very fascinating.
Oh, I mean, this is one of my, this case, like I said, the way it was investigated,